Hey guys, welcome back. I really should have finished the chapter in that last video. I didn't realize I only had about 4 slides left in this set of notes, so we're going to make this really quick. When we're talking about species diversity, we're going to talk about the differences in bacteria, archaea, and how we split them up into different groups, because just like splitting any big group of animals or plants up into separate groups, how you classify that can be a little bit confusing, and there really is a ton of diversity in our microbes. And so one nice piece that we have with splitting these guys up is how far we've come in our ability to separate and magnify DNA in a sample, and we can use that to figure out what kind of bacterial DNA is in a sample. And so we call that metagenomics, and it's assessing microbial diversity by basically analyzing what kind of DNA is from an environmental sample. And we can get a pretty decent picture. If we have a lot of diversity in the DNA and the genes that are expressed, then it's pretty safe to say that we're going to have a more diverse bacterial colony, as opposed to trying to grow all of those individual bacterial colonies separate from one another. That can be very difficult because they don't all grow on the same medium, and some grow more aggressively than others. And so the ability to use just the DNA from a sample does have advantages in that respect. A really cool thing that's going on right now is the Human Microbiome Project. Before we had the Human Genome Project, and that was figuring out all of the DNA inside of a human. And now we have what might be arguably almost as important, what bacteria live on and in all of the humans of the world. It's a huge, huge undertaking, but there are more bacterial cells on and in you than there are cells that make you you. So bacteria play a huge role in our lives, in our immune systems, in our digestion, and it's that way for animals and plants as well. And so we're going to study these microbes that live on or in the human body. So far, we found over 200 species of bacteria just on human skin, and we have found over 1,000 species of bacteria and even a few archaea, which was really surprising, in the gut. And actually, a few of those archaea we found in the gut were even producing methane. They were methanogens, and that's been kind of a funny thing because methane has the ability to absorb and hold a lot of heat. So we blame methane along with carbon dioxide for a lot of global warming issues. And it's been known for a long time that methane comes from combustion, and it also comes from cow farts. So it's been kind of a long-standing joke that it's the cow farts that are contributing to global warming just as much as anything. And now that we know that there are methanogens in humans, we might have to take a good hard look in the mirror at our own farts and how they're contributing to methane in our atmosphere. So thanks for warming up the earth, I guess, literally and pun intended. So sample of bacterial diversity. There are major bacterial lineages. When we start to split them up, we can split them up by if they tolerate heat well or they tolerate salt well. Heat is thermophile. If they love heat, if they love salt, it's called a halophile. And if they make methane, they're a methanogen. So we kind of work with their environments that they live in. And so you guys already know that cyanobacteria produce oxygen because we've talked about that with the creation of the world. And now we've learned that some of them actually fix nitrogen, which is a huge favor to plants. Because plants, there's abundant nitrogen. 78% of the atmosphere is gaseous nitrogen, but it is inaccessible because it is nitrogen atom triple bonded to another nitrogen atom, and that is useless to plants. But these cyanobacteria and other bacteria can take that nitrogen and convert it into ammonia and other nitrogen compounds that plants can use. So they're very, very important there. We also have proteobacteria, which is super simple bacteria that we think have been around for a long time. And one member of this bacteria is so large that we can see it without a microscope. And that's pretty weird because that's not what you normally think of. When you think of bacteria, you think about needing a microscope, but there are some that you can see without them. And I'm not going to try to say that name because it's really something. Helicobacter pylori we've known about for quite a long time. It can survive in some pretty nasty environments, including your stomach acid, which has a pH of about 2. We used to think that stress caused ulcers, and then we found out that actually there's a bacterium in there that can survive your stomach acid eat away the mucus lining, then your stomach digests you, which is incredibly painful, ulcer, blah, blah, blah. And E. coli is the most studied bacteria. It grows on a lot of stuff. It divides quickly. It's a really good bacterium used in the lab. There's a bunch of pictures. You've got your cyanobacteria here. All these are photosynthetic, and then this crazy little guy right here is a cell that is nitrogen fixing, so very, very important. Here's the biggest bacterium. There's its name again. I don't really want to try to say it. It lives in sea sediments, and it stores these white dots that are sulfate compounds, which is kind of amazing. And then we have another one that hunts other soil bacteria and eats other bacteria, so that's kind of cool. And then if it runs out of food or conditions get bad, it makes these little spores, sends the spores out to go reproduce and grow somewhere else. And so that's kind of a unique strategy for bacteria, because we don't really see that whole spore formation and release until we get to things like protozoans, ferns, some higher, a little more complicated stuff. And then we have lactobacillus, which is a bacterium that ferments sugars in milk. It's the reason that milk goes bad, and but we also use it to make yogurt, so it's not all bad, I guess. Some of these will decompose things in natural settings as well, so very important. Major bacterial lineages, we have gram-positive bacteria. Basically, they show up with gram staining. They turn purple or pink. And most of these guys are chemoheterotrophs, so decomposers. They need a chemical. They're eating something that's already made an organic chemical. Lactobacillus, you just saw, eats the sugars made by milk or yogurt. And then there are a few others that are chemoheterotrophs, and these include really small spirochetes and chlamydius bacteria. And these two are unrelated lineages, and they are super, super, super small bacteria. One of them causes syphilis, the other causes Lyme disease. And so there's some other kind of cool bacteria. So then we have archaea. And archaea, in the 1970s, Carl Woese decided that they were pretty significantly different from bacteria. Until then, we just grouped them all together. Archaea live in a lot of the same places that bacteria live, and they have many important differences. Their cell wall is different from bacterial cell walls. The archaea actually have histone proteins that they can wrap their DNA around, so they're a little more organized with their DNA, which is kind of weird because archaea live in super, super violent environments, and we kind of thought they were around before bacteria. Archae, means old. It means ancient life, and so we were kind of thinking that maybe these guys were around before bacteria, but they're also a bit more complicated than bacteria, so there are some minor problems there. Archaea thrive in pretty hostile habitats, really high temperatures for the thermophiles, very salty waters like the Great Salt Lake, in your halophiles. And a lot of these guys make methane as a byproduct. So, points to ponder. Bacteria, viruses, archaea, how does the human body respond if viral invaders or bacterial invaders come into the cell? And the other question is, why do antibiotics not work with viruses? And why are viruses so very difficult to target? Hopefully after this chapter, the second two questions should be very easy. First one's a little bit trickier probably unless you've taken anatomy. So that's a chapter. I hope you enjoyed it. I hope you learned some stuff. If you have any questions, let me know in class and we'll talk about it. So thanks for your time.